Hey,
Hi, Dr. Seneff. How are you?
I'm good. How are you doing?
I'm good. I'm good. I'm very good. What time is it in Hawaii?
6 a.m.
6
Still
a.m.?
dark, as you can see.
Yeah. Is it early for you or not?
Well, 6 a.m. is pretty early, but I tend to get up early. It's not torturously early. It's a little
Okay.
unusual to start a meeting at 6 a.m., but it works.
Nice. And thank you very much for taking time for this little chat. I think you agree with me on many topics, simply just it's so confusing
Yeah.
of like what's happening and why things happen the way they happen. And I just wanted to have the doctors here in the EU who actually asked me a lot of questions.
Uh-huh.
And I think in some of those areas you're much better positioned simply because you worked in environmental toxicology and so on. And this autism epidemics is really hitting hard in Europe and everywhere practically. Where
It's so disturbing. Yeah,
yeah,
I've just been on a mission ever since really 2007 was when I started looking.
where glyphosate is used. And I think after all, as we've been discussing potential mechanisms, we at least have some good ideas that we can
For sure.
discuss.
Yes, I feel
And
like we're closing in on it.
we're closing in on it. And for that matter, I'm contacted. I have a few requests by decision makers in
Okay.
the EU, and it seems that what Bobby Kennedy or Robert Kennedy Jr. is doing is pretty much in the area of deuteronomics, one way or another. Either they say it or they don't. It's not really the prime idea here, but at least their approach is very much like a natural, clear biochemistry is behind all those decisions and especially the new recommendations. And I just wanted to get your idea and find out exactly how we can have these doctors the best way, especially regarding autism and how you see the future unfolding. So, if you don't mind, I just start with the like main question, which is pretty much just like what you've been studying, is what's the current state of glyphosate and links with autism and how those line up with some of the future papers, ideas, and research that you are planning to do.
I think the relationship is extraordinary. I mean, first of all, there's the correlation, which is incredibly perfect. You know,
And
if you
you
look at the
established this back in 2015, as early as
right,
2000.
even to, yeah, 2015, I believe so. Nancy Swanson wrote the first paper. She was the first author on a paper that had all kinds of plots in there, like over
Exactly, yeah.
30, all these different diseases and conditions that were going up dramatically in the United States, exactly in step with the rising glyphosate usage on core crops. And that was just really striking to me. Um,
Mhm.
I was I was convinced really by a single lecture that I heard back in 2012. Professor Don Huber, who is now retired, still active, amazing fellow, and he gave a 2-hour presentation on glyphosate at a conference that I was attending. And when I walked in the room, I didn't know what glyphosate was. When I walked out, I felt like I had solved the problem of autism. Like I really had a tremendous lead to go with. And I went back and started reading everything I could about glyphosate because when he talked about in his lecture and the things that glyphosate does to biological systems and to humans and to the soil bacteria, it lined up so well with what I understood autism to be because I'd been reading all kinds of papers about the complex comorbidities of autism, very complicated disease or condition. And it just looked to me like a slam dunk. I mean, I was just so amazed. And I just dropped everything else and started deep dive into glyphosate at that point.
And what was his take at that point as mechanisms or causes are concerned?
Well, he talked about a lot of things. One thing he showed was mineral uptake into plants was severely decreased in the presence of glyphosate, and sulfur was way down. And that was a big cue for me because I was aware of sulfur problems with autism. That's pretty clear. The autistic kids have a lot of problems with sulfur, sulfur sensitivity, sulfate deficiency, excess sulfur metabolites in the urine. I mean, there's something going on with them that's very fishy with regard to sulfotransferases in particular being disrupted. And then insufficient heparin sulfate in the brain ventricles. I mean, very clear problems with sulfur.
When
in my book, I
go
wrote
ahead.
my book, Toxic Legacy. And in that book, I talk a lot about, my theory about what glyphosate does, which I think I believe it's true. Tremendous pushback. The industry claims it can't happen. I think the evidence is overwhelming, even from their own studies, that glyphosate is getting into certain proteins by mistake as a glycine analog. I absolutely think that's happening. And if you take that assumption, as I did in my book, you can find all these diseases that are going up dramatically. You can find specific proteins that are associated with those diseases that have what I call a glyphosate susceptibility motif. They bind to phosphate at a site where glycine is highly conserved. I think that is crucial. And I think that explains why glyphosate is such an insidious, demonic molecule, because it's a slow kill. It's going into your proteins like bullets throughout your body, and certain proteins get severely disrupted by it. And then they don't work right, and then you get disease. And I think it works extremely well to explain all of those conditions: diabetes, obesity, autism, Alzheimer's, thyroid cancer, pancreatic cancer. It's a huge list.
Yeah, the paper, go ahead.
Well, inflammatory bowel, just think of another one, but IBG,
The fact that, like, what really hit me is Andy Wakefield's paper, the Lancet paper, that was withdrawn, not by him, but by the editors in the pharma industry or the agriculture industry. And there's actually mevalonic acid in the urine of these kids. And I'm a biochemist, so I'm not involved in inorganics as much as I'm involved in organics, because that's typically a TCA cycle abnormality or a very serious mitochondrial defect.
Yes, and in fact, glyphosate suppresses, I think, perhaps all dehydrogenases. And in the TCA cycle, you have a lot of dehydrogenases.
Oh yeah, of course.
Succinate dehydrogenases in particular. There have been papers showing glyphosate suppresses that in mammal studies, possibly even in human studies, I think. Succinate dehydrogenase and also malate dehydrogenase. And what is the other one? Glyceraldehyde. I forgot. I'm sorry, I'm not making that one work. But there's a third one in there.
Yeah, there's a pyruvate dehydrogenase that makes the entry of the acetate into the cycle.
Right.
And that's a thiamine-dependent enzyme. And it was, to me, it was a like huge push on like really looking into this, like the deuteronomics realm of this whole story, simply because if your mitochondria are branching, the TCA cycle is a catabolic cycle with some anaplerotic or anabolic capacities because of the neurotransmitters, precursors, and the urea cycle and so on. But mostly when it's branching, meaning that actually cannot advance, it has to kind of get rid of certain metabolites in and within the mitochondria that appears in the urine, because these are actually water-soluble organic acids. That's a huge toxicity sign right there, which and those levels are really enormous compared to the controls. And we'll cover deuteronomics a little bit later, but it seems to me that such a terrible damage delivered to mitochondria must have a profound effect on especially developing tissues, just like
there's actually several papers that show that glyphosate specifically disrupts the mitochondria. Published peer-reviewed papers.
We'll catch on on this a little bit later because we're going to show your graphs from your paper that the correlation coefficients and those regression parameters, which are actually even scary tight. In biology, anything that is above 0.8 as a correlation coefficient, we actually pay a very, like, strong and urgent attention to it because that means there's a relation between those two factors. And looking at glyphosate application and the autism epidemics and those trends, those are like 10 to the minus 8 or 7.
Yeah, and 0.97
0.97
correlation, I believe, between autism in first grade in the US and the use of glyphosate over the previous 4 years, integrated over the previous 4 years.
Those are very insightful graphs and very important data that I'm showing to these doctors and also to some decision makers because this has to be dealt with one way or another. And we know what the consequences are. So this is practically a very important work that Bobby Kennedy or Robert Kennedy Jr. is taking on. Have you heard of Trump actually signing a executive order that the auto pen used by Biden and Fauci's clemency is void? Or,
yeah, I have heard that. Yes,
you heard
that's interesting.
What's going to be the outcome of that?
I don't know. I don't get enough involved in politics to be able to predict these things. They're so extraordinary things are happening in politics right now in the United States, and lots of people are working hard against RFK Jr., which really frustrates me because the pharmaceutical industry has a tremendous amount to lose with the agenda that RFK Jr. has in mind.
Yeah,
and they know that, and they are fighting so hard.
Yeah, they want him out simply because for them that's life or death question.
Right. Well, they'll lose a huge amount of money,
or
maybe even go to jail. I don't know.
Oh yeah. And for that matter, I think Bobby's book about Fauci is pretty revealing of this whole business that he was doing. And what is your take of that material or that book?
Yeah, or even that topic. I mean, the whole COVID story is really bizarre. And I early on, I predicted that the whole game really was to get the mRNA technology out the door. They saw it as a tremendous opportunity. Now, you're going back to the origin.
Sure.
And I think it's pretty certain at this point that its origin is that laboratory in Wuhan. And then a question of what were they doing, why were they doing it, comes up, and that's not clear either.
But what's really interesting is that back in 2020, early March or mid-March, we were going to go and eat dinner with Bobby, and I sent him an email. This whole COVID is just a baloney. It was right after the lockdowns, and
good for you. Wow, I didn't know that.
I wrote him an email that this is a baloney, and we had to postpone simply because he didn't want to have dinners at that time because nobody knew exactly what is happening or what is going on. And I think his kids, because they are affected by some of those vaccines, I think for him it's also a very important agenda just to go after all these vaccinations and Fauci's work and find out what those ties are with pharmaceutical industries and how those play out after all. So
and really what I think is that they were determined to get the mRNA approved. If they could get the mRNA technology approved in those COVID vaccines through emergency conditions and skip a lot of steps in the evaluation process, I think they understood that was the only way they were going to get that technology approved as a whole, the mRNA technology.
Exactly.
Which they desperately wanted for their cancer, all their ideas about using it to treat cancer.
Yeah. And as we've been involved in this research for a long time and we wrote papers about this, we know in depth how harmful these technologies are in fact. And now I just learned from the Washington Post that the FDA is implementing new regulations simply because children died of the COVID vaccines. Right. And I think the head count is where they can actually prove and cause the, like 10 or more than 10 children. So now Dr. Washat is now, he's changing the whole FDA's agenda behind these.
I saw that. That's very interesting.
And what about nutrition, the ultra process? What is your take on combined effects with glyphosate?
Well, for sure. I mean, the ultra processed foods are very deficient in lots of interesting micronutrients, beyond just minerals and vitamins. There are all these, complicated molecules that are produced by plants, for example. So when you eat plants, you get a lot of molecules that you wouldn't get with the heavily processed foods. They're basically just sort of sugar and fat. I mean, they're just very plain ingredients in those. They sort of think of just the basics of the and of course the foods the crops that go into those processed foods are mostly the GMO Roundup Ready crops. You have the corn, the soy, the canola oil, the sugar beets. So you've kind of got the basic ingredients that you can use to make all kinds of cookies and candies and whatnot, and soy protein bars, And so certainly those are all GMO Roundup Ready. So you've got glyphosate all over the food supply. Zen Honeycutt, my friend Zen Honeycutt, who's the founder of Moms Across America, has tested. She has had her moms get samples of school lunches at the public schools and also samples from fast food restaurants and send them off for glyphosate testing. And in the case of the fast foods, it was 100% of the samples came back positive, and almost 95% came back positive for the school lunches. So it's all over our food supply. The government thinks it's safe. They've been fooled by the industry to believe that glyphosate is safe. So they know it's all over the food supply and they don't care. They don't even measure it.
Yeah, because they are not responsible for anything. It's practically the worst situation for population disease processes when there's nobody responsible for this and politicians approve it. So after that, it's practically a circle of a, like, just like a death circle for that matter. And there are diseases just popping up. And I really fear that our younger generations are truly exposed to extreme dangers simply because of the glyphosate and because of the food products and the ultra processed foods. And this is why I'm so happy you are first helping us with putting these big pictures together, and we can actually go into details as biochemistry is concerned. Your newest paper is a continuation of the work with the German investigators, or what's the summary?
I don't know which newest paper you're talking about. I have several in the pipe.
Heiko Schöning was just writing back to us
with
because of them.
And
with
Maria,
Falco,
yes, the three of us collaborated on this paper, and Falco was the lead on that. It's an amazing paper, I think, and we've published it as a preprint. And it's on the lipid nanoparticles themselves, so it doesn't concern. It mentions the spike protein, but it's more focused on the lipid nanoparticles themselves, and to consider whether they might be toxic, and of course, why they're designed the way they are. It's a review paper, a very long review paper with several hundred references, and we have submitted it to a journal, and we're waiting to see how that goes. But it is a preprint. It's available as a preprint. And
how would you sum it up? What's the toxicity there?
Well, we showed that they clearly disrupt signaling, and particularly there's an interesting aspect to them that they have these I'm losing the word, phosphatidyl inositol, which gets phosphorylated in all kinds of ways. With 3,4,5, it has all these phosphates that can get attached to it, and there's a tremendous signaling process that goes on with those phosphates, putting them on, taking them off in the cell. They're very powerful signaling molecules in the cell, and they are present. There's versions of them that are normally in the lungs that are present in the vaccine, that would have unpredictable signaling consequences. We're mostly talking about hypotheses that have not been adequately investigated with respect to the potential for them to cause trouble. And then there's a lot about the signaling and how they induce, a inflammatory response, and then all these cytokines and all of that, which is known. That's their purpose in a way, and of course the cationic lipids, the concern about those, disrupting endosomal processes, the lysosome.
What's really interesting, what's really interesting about this paper in particular, when I was at Ohio State with Dr. Patricia Chauvin, we were actually working on a lung injury, an acute lung injury protocol. And actually, we induced that with lipopolysaccharides, meaning that it's actually so toxic that actually it causes in experimental animals in certain dose multiple organ failures. And those are actually shock or ARDS or coagulation, and you just name it. It's the most toxic material, I would say, that you can inject of a biological origin into an individual.
And another thing that's interesting, and we didn't write about this, but I am quite fascinated by this idea of, you may have thought about this as well, cholesterol itself is derived from 3 acetate molecules. And I think acetate is going to be a low deuterium nutrient if it's come through the microbes. The microbes recycling hydrogen gas, I think, is absolutely critical for the production of deuterium depleted nutrients for the host, one of which is cholesterol. And when they put cholesterol in those mRNA vaccines, it's not deuterium depleted. And it might even be, there's there's material on the web that suggests that it would be good to dope the RNA with deuterium to make it stronger and less able to break down. There's a company that promotes that.
Yeah, and they actually use heavy water for,
Exactly.
And we actually cite this in our paper, these protocols, which are very, very dangerous, adding deuterium or
if they do
changing.
That, it's very serious. And we didn't talk about that at all in this paper because it's too, speculative, but I would love to know whether they actually did enrich them with deuterium, but even just making cholesterol in the laboratory, and not just cholesterol, by the way. That's just one of the things, one of my new papers is looking into synthetic versions of molecules that are known to be biologically beneficial that people take as supplements. And so, for example, choline bitartrate, and then what's it? Carnitine, L-carnitine, choline bitartrate, and methionine, S-adenosine methionine, SAMe. Those 3 are popular supplements that people take. They're in some of these men's multiple vitamin and mineral pills. They're considered to be healthy. They're choline and carnitine, as you know, are present in animal-based food.
Of course.
And they're important because they feed into the methylation cycles.
Well, there is a French team of investigators. They can actually measure deuterium enrichment in certain carbon positions in any organic molecule. And
that's fantastic.
And when they measure, and the study they did was a capsaicin. It's available from plant source and also available from pharmaceutical companies. And the difference in deuterium content was some 20 ppm of the artificially made.
Yes, that's exactly what I'm thinking.
Supplement about 20 ppm higher.
Yes.
And actually, I was thinking about this, that any supplement you take or any organic compound of a synthetic or laboratory or industry origin, because it doesn't have the natural deuterium depleting pathways or mechanisms that are essential for metabolic functioning and for the absorption and the biological function of those chemicals. Because of the heavy deuterium load, it's practically, first of all, it's useless. Second of all, it becomes toxic. So they don't really deliver the biological function. If you want me, I can send you those papers where you can actually.
That would be great. Yes. I mean, I discovered there's several papers that are looking at things like L-carnitine and and choline as supplements. And very fascinating, because these papers are comparing, for example, choline bitartrate, synthetic choline bitartrate with natural phosphatidylcholine. And when people were eating lots of eggs, no problem. But if they took this supplement, they got elevated levels of TMAO, trimethylamine oxide. And that TMAO is a very interesting molecule. I've been reading tons of papers about it recently because it is associated with all kinds of diseases. I think it's a marker of excess deuterium load systemically. And it comes about because of the microbes. The microbes make the TMA, and then they metabolize it, and they feed it into the methylation pathways. But those microbes are anaerobic. When you have inflammation in the gut, they don't thrive. The TMA doesn't get cycled into the methylation pathways, and instead goes to the liver and turns into TMAO. It's an indicator of TMA that's not being processed. First of all, because those microbes are sick because of the inflammation. And secondly, because the enzyme itself that does it has a very high deuterium kinetic isotope effect. So I think it may be a mechanism the body uses as a sensor to ring alarm bells that the mitochondria are in trouble with too much deuterium, because the microbes provide so much deuterium depleted nutrition to the host.
I think that's the function of microbiome, for that matter.
I think so too.
Separate is to protect the nanomotors. That's the ultimate goal of all these biological function and some other enzymatic mechanisms in and within these cells, the mammalian cells, the host cells. And
it's so fascinating, because the microbes actually don't mind if they are loaded up with deuterium. So they're basically producing deuterium depleted nutrients for the host while they're concentrating deuterium internally.
Yeah, because
and that all works.
For their clonal expansion, that's necessary to keep their DNA unstable, so they can actually divide continuously and
even mutate, which was really interesting. I just learned that from our crew recently. That was so fascinating, that the excess deuterium would facilitate mutation, which would allow them to adapt more quickly to environmental exposures.
Yeah, and I think this is why polymerase theta is so important for these prokaryotes, because they don't have to break down their RNA. They can actually, after the ribosomes release the RNA, they can recycle them back to DNA. So that's practically a good enzyme to salvage a big step in de novo nucleic acid synthesis. And that's how
that is very interesting.
Yes. We will discuss this in the newest papers that we are writing. So I'll update you on these topics a little bit. So this is one of the dangers of modified mRNA vaccines, is practically we still have this enzyme in our mammalian or in our host cells, and especially muscle cells that are not dividing, but they have DNA. Actually, they have aneuploid DNA, for example, neurocytes. They actually might use this enzyme simply because there is a template, because our messenger RNA has to break down very quickly. We can't modify it. So that's why I think, after all, the biochemical mechanisms are all there to explain some of those side effects and consequences of these very toxic treatments, after all. And now we see the impact of this as public health. There's a German paper out there showing the excess mortality is increasing everywhere, all-cause excess mortalities. And Peter McCulloch writes very intensely on these topics, simply because
he's very passionate
about it and so on. And for that matter, our deuteronomics discussions, I really enjoy the current.
It's been so fun. I really appreciated being a part of this incredible team.
Because Roman Zuberev from Karolinska, he's the physicist and the analytical chemist behind of all the data that he cannot explain from the heart, simply because he's involved in different areas. But we as biochemists and scientists, we have a kind of a better insights of really truly what happens if they measure some of these extremely different deuterium levels in certain cell compartments. And I work with a university here in Hungary, the geophysicist, and they say that it's not that deuterium is increased, but the level of those changes or increases to serve the phenotype of that sort for them is really truly surprising, simply because in the environment you would not see those deuterium levels anywhere in.
Oh, but they can adjust, you mean? I know I've seen that they can make E. coli. They can sort of slowly move them towards the goalpost of extremely high deuterium, and they can still survive through their adaptation.
Yeah,
process.
it's an adaptive process, and it seems that deuterium is everywhere for that matter.
But,
it's just
and
practically
it facilitates the mutations that allow them to adapt, which is really quite interesting, you know, because it increases the risk of DNA.
It's more like a circulus viciosus. It's practically just more deuterium, more mutations, more adaptive capacities or capabilities in biology.
Yes, I think but it looks to me like the microbes, we know they can adjust very quickly because we've seen that in experiments.
Oh,
and
yeah.
Maybe part of the reason why they can is because they hoard deuterium.
Oh yeah. They actually retain deuterium and they pump hydrogen out. So it's for them, because those ATPase pumps can be used two ways.
Right.
The bottom line is deuterium cannot pass through those nanomolar.
I know that you gave me that paper that was so fascinating about the E. coli and the VATPase pumps,
Yeah.
And I remember the quote that deuterium perhaps not at all, because the authors
replace.
They can't get through. And that's so interesting because actually those pumps are used to acidify the lysosome, turn the endosome into a lysosome. They pump, and we have those enzymes as well. And they want to put low deuterium hydrogen into the lysosome because that's going to hydrolyze all these complex molecules into individual small molecules, putting hydrogen atoms on them. It wants them not to have deuterium. So it wants to have those lysosomes be low in deuterium as well as the mitochondria. And then when you get cancer cells turn around and take those VATPase pumps to the membrane, and they pump the low deuterium protons out into the environment. Yeah, they're actually supplying the immune cells that have invaded the tumor environment. They're supplying them with low deuterium water.
And that's why I put together this mitochondria user guide, because it seems that mitochondria carry those 13 gene proteins that cannot adapt to anything because there is no male alleles in the system. So it's practically a maternal DNA in the mitochondria that determine the protein size and the protein structures that supply only protons for the intermembrane space by NAD pH or NADH dehydrogenases, and also the stator part of the nanomotor is carried, that gene is carried by the mitochondria. It wouldn't give it to anyone or any other cell organelle.
It's their own private DNA.
It's only private DNA. Mitochondria has DNA. It carries.
I know it does. Yes.
Defined 13
and the mRNA
proteins.
The NAD dehydrogenase is a mitochondrial gene.
Yeah, and so is the stator. The stator, there's a rotor and a stator in the nanomotor. The stator that supplies the protons for the rotor, that's also mitochondrial
so interesting. Gene. That's really fascinating. So the mitochondrial genes are really focused on that low deuterium problem.
Well, it's cloned ever since.
Yeah.
Practically because it's only the mother carries those genes, because at the time of fertilization, when the zygote is formed, all male mitochondria are destroyed by the trophoblast layer. So there's no male mitochondria or DNA that gets into the mother's mitochondria. If there is any mutation in any of those 13 proteins, those are lethal. Meaning those definitely prevent reproduction. Meaning that
because they're hanging out in the mitochondria, they can't mutate very fast, right? They're protected from that because they don't have a lot of deuterium.
And there is no allelic variations either, because there's no male alleles to this. So
I see, that's so interesting. That's very insightful, and I didn't know that. That's
so even evolution has no touch to it, or even evolution has no access to it. So your mitochondria will not be able to adapt to anything other than the proton itself. It can adapt to a higher deuterium containing diet simply because those proteins will just break down or they just stop functioning. So for that matter, mitochondria is really truly very interesting for them.
They're so fascinating. I'm just thrilled with the discovery process, and I'm so delighted to be part of your
well, we all add our own parts,
we all have different insights,
It's kind of like a family game of everybody has good cards, and we play when we have to king up and prove and show something really fundamental. And I also wanted to ask you if the glyphosate, again, the glyphosate story, as it goes back to the mitochondrial damage, which is mostly my area of expertise, the TCA cyclin, the proton transfer, and so on. Glyphosate also compromises, I believe, oxygen delivery to the mitochondria. And simply the fact that the red blood cells have these hemoglobin proteins that bind oxygen, and between carbon dioxide and oxygen binding, there is a glyphosate or glycine or glycerol 2,3-phosphoglycerol that regulates this oxygen and carbon dioxide binding to the molecule. And I believe glyphosate can actually replace this 2,3 glycerol molecule, which is the product of glycolysis, which red blood cells use. And glyphosate, for that matter, compromises oxygen delivery that actually makes this whole scenario even worse simply because there is no oxygen for low deuterium or deuterium-free metabolic water formation in the mitochondria. And this was...
Glyphosate also suppresses an enzyme that's the first enzyme in the synthesis of that chiral ring that is in the hemoglobin. It disrupts the synthesis of hemoglobin at the very beginning. Because it's a glycine analog, but the phosphate, you make a very good point, because phosphonate, which is attached to the glycine molecule in the methylphosphonate unit, is probably works as a phosphate analog. And so anytime phosphate's involved, glyphosate could even be binding to the phospholipids in place of phosphate. I don't know. That's possible, I think.
I see. And it's just so interesting, and we'll continue on these topics simply because there's always something new insights and we can use for these arguments. But definitely this is one of the most toxic scenarios that you can encounter in the environment.
Yeah, and of course it's well known now that glyphosate disrupts the gut microbiome, and it preferentially kills off the beneficial bacteria and allows the pathogens to overgrow, and then that brings in the immune cells, which causes the inflammatory bowel disease. That's are you okay? Yeah, you're there. The inflammatory,
I'm
IBD,
here. I'm here.
That's an epidemic right now in our country, and the inflammation is what's disrupting the anaerobes. The strict anaerobes are the ones who do the hydrogen cycling. And the hydrogen cycling is so important. The hydrogen gas, molecular hydrogen, is a critical part of the process by which they scrub deuterium from those organic molecules.
Yeah, so and this is how our retina works. It's simply there's no blood flow, but there's water that is broke by light. And this hydrogen and oxygen gas can supply the receptors, the photoreceptors in our eyes. So there's not blood needed for this whole process to power. And they produce the most ATP, but it seems like they do this without a carbon source. They do this just simply breaking water by light.
That's so interesting.
It's really
that's fascinating.
And what's even more interesting is there was a German hospital, and they were actually using 670 nanometer red light for 3 minutes exposure, and it improved color vision for 2 weeks by about 20% if they did this in the morning while the patient was in a low deuterium ketosis. Once they ate their lunch in the hospital or the breakfast, and they did the experiment in the afternoon at 3 o'clock, it didn't work because they were out of they actually were kicked out of their deuterium depleted water producing ketosis after night sleep. So
That's
practically the
amazing.
experiment didn't work. So there's a lot of connections here that we can
Yes.
use for this hydrogen gas to be such a fundamental,
It
um,
really is. I really
critically
have come
important.
to believe that. And it's really interesting with the methylation cycle because that's something I've been
And only
studying.
the gut microbiome can produce that simply because those
Yes,
bacteria are capable of producing hydrogen gas while they are
exactly.
retaining deuterium for their own purposes.
Exactly. And we don't have those hydrogenase enzymes. It's so amazing.
Yeah, yeah,
We depend upon them for that.
yeah.
And
It's
it's
really, really
crucial,
interesting.
absolutely crucial. And of course, the short chain fatty acids, butyrate is another one that's become a hot topic. And that's
I
one of the
see.
short chain fatty acids, low deuterium. Butter, of course, is a
Hmm.
low deuterium
Hmm.
nutrient, which has butyrate in it.
Oh, yeah, yeah, yeah. If it's
And
natural
that,
grass fat source.
yes, I know. Well, that's right. That's the other problem is you have to
And
be careful
even
with,
it's really interesting because we actually measured in Holland at the Vrije University butter that was from local suppliers, livestock, and it was 137 ppm, while the butter from Hungary, from a different pasture, was only 110 ppm. So we're wondering
interesting.
what's the difference of that 25 or 20
That's pretty
ppm,
huge.
which is pretty huge. Now, it's really interesting. Actually, in Holland, the grasslands are below sea level. So there
Mm,
is no fractionation there. And they also feed them because they don't have natural like grasslands. They have to
mm.
use a seed or a grass that is not natural for the habitat of the cows. And you see, if you don't feed them with the right pasture or the right source of their plant that they are eating, their deuterium regulation is totally off the scale, meaning
Well,
that
here in the US, it's got to be terrible because we feed our cows corn and soy,
corn, yeah, the corn is the
GMO
worst
Roundup
simply.
ready.
Yeah,
Yes,
yeah. GMO is there, Roundup is there, corn is there, sugar is there, starch is there. You are just asking for it.
yes.
It's just practically just asking for it. And when you combine that with the number of vaccines they are getting and
Oh, I
prepared
know.
with heavy water and with chemicals that are really truly bypassing the deuterium scavenging or guarding mechanisms of the microbiome or the cavity, the oral cavities, epithelial cells, and inject these into the muscle, that's practically just a toxic environment
And
for
what's interesting
cancer.
too is that glyphosate is a contaminant in the vaccines. I think you know about that, right?
Oh
This
yeah,
is the
yeah,
work of
yeah. Oh yeah,
Anthony
yeah.
Samsel and Zen
Mm-hmm,
Honeycutt. Each of them at the same
mm-hmm,
time independently
mm-hmm,
sent several different vaccines off for testing and came back
mm-hmm,
positive
mm-hmm,
consistently with the live virus vaccines, and
mm-hmm,
particularly with the MMR vaccine, by the way. That one had by far the highest levels of glyphosate
mm-hmm,
compared to all the other vaccines. And it's because the live
mm-hmm,
virus is grown on nutrients that are derived
mm-hmm.
from these GMO crops that are contaminated with glyphosate. So it gets into the vaccine, and that's really
Injecting those
terrible.
into the muscle, that's just
It's
practically just
much,
asking
much
for it.
worse to inject glyphosate past all the barriers than to eat it,
What do you think what the ACIP will recommend this week? Because they are working on the childhood schedule, I
I'm
believe.
hoping they will get rid of the mandate that the child gets this very toxic hepatitis B vaccine at birth. That is so crazy. And I
Yeah.
know they're bringing that one up. They're focusing on that one as being
Mm-hmm.
uh,
Mm-hmm.
not necessary. It is so
It's
ridiculous to me.
senseless. It's
Yeah,
senseless.
it's
It's
just
ridiculous.
crazy.
It's because nobody is responsible for it. They can do anything they want.
And it
And it
has a
just
high
makes no
level of
sense.
aluminum, of a toxic form of aluminum. It's just like, why would you
Oh
do
yeah.
that
Oh
to a
yeah.
newborn child?
Mercury and aluminum. We found vaccines, they have both. So it's just aluminum and mercury. That's
Mercury is in some of
practically
the flu vaccines, I know,
there
but
you go.
I don't think it's in the
Yeah.
Hep B, just the aluminum.
And I think
Um,
they are changing the flu vaccination schedules as well. I think there was
yes,
some.
and I think they're going to try to get rid of. I think that it's aluminum that's in some of the flu vaccines. It gets confusing between the two. I studied this at the turn of the century, 2000. or the issues at the turn of the century, when they claimed to take the mercury out of the vaccines, and then the autism rates continued to go up, and they said, ha ha ha, mercury doesn't cause autism. But they introduced vaccines that contained a lot of aluminum in a toxic form of aluminum at the same time as they removed the mercury. So they just transferred it from the mercury to the aluminum. And then
those are, they still give them to mothers, and they still give them to children, and so on.
Pregnant women, which I find horrific, they give them these vaccines that contain
and the half-life of aluminum materials is like 7 years in the human body. So the biological half-life, meaning that they just never clear from the system.
And glyphosate binds aluminum. Glyphosate binds aluminum and wraps it. 2 glyphosate molecules will wrap around an aluminum atom and hide its +3 charge, which makes it into a small non-charged molecule that makes it much easier for it to get across barriers. So if you've got glyphosate in the MMR vaccine and aluminum in another vaccine that you're taking at the same time, those two can collaborate.
They collaborate, and they don't clear biologically, so they add up after all the toxic effects. Synergy. I wanted to ask you 2 more questions, and I will thank you so much for your time and giving us this enlightening conversation. One is that what is your take on establishing a National Deuteronomics Institute in the United States?
I would love it. That would be so fantastic.
I think we are there at that point because of the role of deuterium in so many biological processes.
It is essential. It's frustrating how little we know about deuterium and how few people are studying it. I feel like I want to sort of shout it to the rooftops because I think it is so important and so central. And I'm trying to get as many people as I can, get their attention, through podcast interviews and whatnot.
We have a lot of scientific as well as like really more like as considering the future of our nation and the young kids and our public health issues. I think deuterium is significant enough to have its own institute among the others.
Certainly some kind of initiative to fund research on deuterium would be fantastic, coming from the government.
We will go with this to Bobby once they have the midterms. So, and the last question I wanted to discuss with you is that the origin of species or deuterioms, because it seems like
I love it.
Because I mean, as serious as it is, even Roman Zubarev, who measured those very high deuterium in collagen, he called it abnormal in his paper, in the title of this paper. But it's not abnormal. It's practically what the physiological pattern of a deuterium or deuterium in that species, simply just to make it able to adapt to the environment. And
is this the seals that had the high? That's such a great paper. I really enjoyed that, and it was very insightful. And I think we also probably concentrate deuterium in our own collagen. And sportsmen, people who actually exercise more or actually use their joints, and they have to be a cartilage that is collagen stronger. That's the same with animals with big, large body grass-feeding animals. And it's funny, it is. My friend, he herds water buffaloes in one of the Hungarian mountains, and the wolves, they take some of those cubs down and they eat the interiors, the microbiome first. That's the first what they eat, and they also eat the cartilages of their joints. Oh my, that's so interesting.
They leave the rest of the carcass for the vultures. But they actually take care of the joints, the cartilage, the collagen, and they eat the interiors because that's where all the deuterium depleting microbiome forming bacteria are.
That is really amazing.
And that's why I figured microbiome is just as important defining a species like the number of chromosomes, because if the chromosomes are different in number, they will not able to propagate, but as long as they are the same, number, their weight and their size have to be the same as well. And that really depends on the deuterium load in those chromosomes. And I think the infertility epidemics in the US and in the Western world is simply they don't eat the same food, the husband and wife or the young couple. One eats at a McDonald's, the other one eats salad in the workplace. So their chromosomes are different load with different deuterium loads, so they are not compatible.
Wow.
And I think in the animal kingdom, since actually the female fetus, they close in their follicle and their eggs by the first 3 months of their intrauterine life.
I remember reading about that. It's so amazing that the egg matures. The next generation is already getting ready in the fetus.
And the mom is in ketosis in the first three months simply because they just have morning sickness. So it's practically a low deuterium. Yeah, I think nature already figured it out. And when they when they grow up and when the mating season comes, the males, since they produce sperm continuously, so their chromosomes can be loaded with deuterium. For the mating season, they have to stop feeding and they have to do these mating fights. And they actually just test their nanomotors and deuterium levels, who is stronger, who is more determined, or they have to do these mating dances where the female can see how fast they move and what kind of turns that can make. So it thinks it evolves all around deuterium, how the males can deplete their deuterium for the mating part, and that's biology has all these tricks to do to be able to measure this.
It's fun to look at those things from this perspective of deuterium. I've been doing that with metabolism to take a look at the mitochondria and what they're doing, even like oxidizing cholesterol inside the mitochondria, and making deuterium depleted water in the process. That the water is sort of ignored by the biologists, but that's the important product.
Oh yeah,
all these lipids.
It produces most of the heat that we produce. It's 10 times more than what ATP carry. ATP is needed for working proteins, but the formation of water, the joining of hydrogen and oxygen produces 10 times more by kilojoule per mole energy than ATP carries. So it's very important for our heat production, and it's part of the energy production process and so on.
Yeah,
and
it's all very fascinating.
And I think after all, because the males have to stop eating and they start keeping fighting and exercising, so they get into a deuterium depleting exercise ketosis. That's the key for successful fertilization.
Yeah, and if they're strong, that means the deuterium is in their bones and in their joints instead of in their mitochondria. So whenever you concentrate deuterium in one place, you're depleting it someplace else.
Somewhere else, yeah. That's what makes strength, and that's what makes durability so obvious in biology, a very important mating or driving force for the species to survive. That's why I suggested this new direction. Dr. Seneff, thank you so much for your help.
This was fun. I was delighted to participate with this conversation. And I learned more about deuterium.
Yeah, we'll continue discussing this, but I wanted this to kind of have this so doctors in my circles can also have a thorough insights of where glyphosate is, where autism is, where nutrition is, where ultra processed foods are, and the rest we will figure out for ourselves. But this is very, very helpful. Thank you so much, and have a wonderful day. It's actually, the sun is getting up in Hawaii.
It is. It's beautiful. There's a sunrise.
Which island are you in?
Kauai.
Kauai. Okay, it's close to Hawaii.
If you can see.
Nice. Beautiful. And it's close to Maui, I believe.
Kauai is on the furthest west, and then the Big Island's on the furthest east, and in between are Maui, Oahu, I think, and then Maui. I'm not. Yeah, I think it's Oahu, and then Maui, and then the Big Island.
We'll do our geography. Thank you so much. Have a wonderful day.
You too.
Thank you, Dr. Seneff. Good to see you too. You
too.
Thank you. Bye.